Emphasis of this lecture series will be on skeletal muscle, as you will learn more about cardiac muscle and smooth muscle when you learn about the histology of human organs. I'll begin by describing the organization of a skeletal muscle. I'm going to use this biceps brachii muscle as an example, since it's a muscle familiar to us all. For those of you with a knowledge of anatomy of the arm, you will know this is the short head of biceps with its proximal or superior attachment on the coracoid process of the scapula. And this is the long head of biceps with its proximal or superior attachment on the supraglenoid tubercle of the scapula. The distal attachment is down here on the radial tuberosity and the bicipital aponeurosis. The next image is a model interpretation of the structure of a skeletal muscle, seen viewing a section cut through a muscle such as this biceps brachii as the example, perhaps sectioned somewhere across here. The whole muscle mass is wrapped by a thick layer of connective tissue called the epimysium. This is then continuous with the fascia and the tendon attaching the muscle to the proximal bony locations on the scapula and the distal attachments I pointed out on the previous image. Most muscle contains these many bundles of muscle fibers packaged in a thin layer of connective tissue called the perimysium, which also carries blood vessels and nerves to supply the muscle fibers. And within each fascicle, each individual muscle fiber is surrounded by an external lamina and a delicate connective tissue covering called the endomysium. Let's now look at some real muscle tissue at the gross anatomy level. Examine a piece of steak carefully next time you purchase some at your local store. Skeletal muscle is attached directly to bone, as you see here, via attachment with dense connective tissue, but also via a tendon, particularly muscles that attach to our appendicular skeleton and move our limbs. You just saw that when we looked at the biceps brachii muscle. These voluntary muscles move the body and maintain posture and body positioning. Now in this image, there are a number of different muscles, each separated by a thin collagenous epimysium. In preparing histological sections of muscle, the emphasis is on collecting muscle tissue to view details of the skeletal muscle fibers. So when you examine these small tissue sections with the light microscope, you do not normally see this covering. It acts to separate muscles apart so they can contract or relax independently. In many places, there are accumulations of adipose tissue, which are fat cells or adipocytes. Now in the next image, I'm going to show you a cross-section through a small piece of skeletal muscle such as this small region here, with a bit of the epimysium to show the histological details of the relationship between the muscle fibers and their connective tissue coverings. The tissue has shrunk during fixation and opened up spaces in the connective tissue seams. These white spaces are purely artifacts, but they do make it easy for you and I to trace the locations of connective tissue, and in some cases their contents. The muscle has been cut transversely, and so the muscle fibers are seen in a transverse orientation. The epimysium is this thick outside layer of connective tissue along this one corner of the tissue. Thicker seams of perimysium divide the muscle into fascicles or bundles, and provide a pathway for arterioles, venules, and nerve bundles, which you can just see here, even at this low magnification. Its type 1 collagen fibres and scattered fibroblasts, or at least their nuclei, are recognisable. Remember, fascicle refers to a bundle, and as you can clearly see in this image, a muscle can have fascicles within fascicles, each surrounded by a thin seam of perimysium. Every individual muscle fibre with peripheral nuclei present will be wrapped in its own very delicate connective tissue layer of endomysium, too fine to see in this image. This layer would contain many capillaries running in the longitudinal direction of the muscle fibre and reticular fibres. I'd have to look very carefully to find these capillaries, and you'll not see the fine reticular fibres no matter how closely you look, so I haven't bothered about showing you this detail. But remember, it is present and it is important. This structure here, by the way, is a small branch of a peripheral nerve wrapped in a perineural sheath. You can learn about peripheral nerves in the next major lecture topic in this series.